after we decide to become a neuron and we grow our axon out, we'll also be developing dendrites. So the dendrites are going to be the receptive end of the neuron, and they're going to contain the majority of the postsynaptic sites. The purpose of dendrites is to cover some sort of area, for the most part within the central nervous system, but some dendrites exist out there in the periphery as well, but to provide fairly uniform coverage of some area to not overlap with some of your neighbors, but maybe overlap with others, and not to do too much crisscrossing of yourself, so that you spread your dendrites out rather than collecting them into a lump. Now, different neurons are going to have different degrees of dendritic complexity. Some neurons are going to have more of a pyramidal shape where they extend one dendrite up and then have a few basal dendrites, and others will be a little more stellate. Some will have very simple dendrites, others far more complex dendrites. The complexity seems to be encoded genetically, and of course the development of dendrites is going to be guided by extracellular cues. Now the good news is a lot of these cues are the exact same ones that guide axonal development, and this allows one cue to control both dendrites and axons. Usually what it will do is have opposite effects on dendrites and axons for the same cell, but have opposite effects on the axons from different types of cells, meaning that it'll provide repulsion for neuron A's axon, but act as a guidance cue for the axon of neuron B, so that neuron A here grows its axon away, while neuron B, way over on another part of the nervous system, will grow its axon toward A and form a synapse. We'll see a couple examples here, and then we'll talk about some of the differences between the microtubules that we find in the dendrites and the axon. Because when the neuron is first developing, it doesn't actually know which part to make dendrite versus axon. It creates a bunch of simple neurites that could be axon or dendrite, but only one will eventually become the axon. And part of this process is by modifying the microtubules there so that they become far more stable. We're going to need stable microtubules so that we can transport our intracellular components over long distances, because we're going to have to take microtubules if we're going to transport proteins and organelles down the axon. And remember, axons don't make proteins, so we will have to transport them. Okay, so the general logic of dendrites is to cover some area uniformly, to make sure that we're covering all of this receptive area. A good example of this would be the retina, for example, where we need to have uniform coverage of dendrites so that we can have a uniform visual sensation, so that we can pick up light all over the retina. There's two kind of basic principles that we're going to be doing, and that's spreading out our cell bodies and also spreading out our dendrites. So the dendrites from each individual neuron are going to tend to repel themselves. That's like repulsion. And what that's going to help do is prevent the piling up of dendrites into one area, so that as a dendrite grows out and it encounters its neighbor, rather than growing along one another and covering the same area, instead those dendrites are going to turn away from each other so that they cover different areas. And all dendrites are going to do this, and even as they branch out, we're going to have like repulsion so that we get fairly uniform coverage and not a bunch of dendritic fasciculation. So the idea is to make sure that anywhere where we stick an axon, we're going to have a postsynaptic target here. We'll talk about how like repulsion works in just a little bit, but here you can see the expression of GFP in a subset of cells within the cat retina. So they use the Thi1 promoter. This is a neuron-specific promoter, but it's not used in every neuron. So you only get GFP expression in a subset of retinal cells there. And what you can see in the data or in the little cartoon below is that we have relatively uniform distribution of dendrites. And that's what we call dendritic tiling, where the dendrites repel themselves in the same cell and they occupy some little area. They're kind of like little tiles that you can arrange down to form a floor. The mosaicism that we see is the even spacing of the cell bodies. So mosaicism and dendritic tiling are going to work together to make sure that we have uniform coverage. Without mosaicism, two neurons might end up migrating right next to each other and then they're doomed to overlap. So we prevent that with mosaicism, make sure we spread out our neurons enough so that whenever they grow out their dendrites, they're going to occupy their own little space and form their unique little tile. So we got to spread out the cell bodies, we got to spread out the dendrites. Now there's many different types of neurons, and some neurons might tolerate growing next to a different type of neuron. For example, if you have excitatory and inhibitory neurons, it can be okay for them to occupy the same space because they're likely dealing with a different type of information or having different effects on some process. But your pyramidal neurons, for example, which are all going to be excitatory, you want to spread those out and have fairly non-overlapping coverage. Or your Purkinje cells in the cerebellum, same function, so you want to spread those out. So this tiling can be homotypic, where the same type of cell is going to exhibit tiling, but they might tolerate a different cell type living within their tile, or it can be heterotypic, where different types of neurons repel each other. So it's not always the case that every neuron occupies its own little tile. There can be overlap. So what this cartoon is showing us down there on the bottom is the purple is fine living with the oranges, but the orange neurons there, which are a different type, don't tolerate that. Now this tiling is of course going to be achieved by the interaction between surface proteins. So the ones that we need to know about now are going to be tricornered and furry. Furry is just an adapter protein that holds tricornered in the right place. So scattered along the dendrites here, we of course have surface proteins. And what tricorner does is cause turning. So you can see dendritic turning in this time-lapse data here. So 0 minutes, and then they're capturing it 4, 12, 16, 20, 28. So they're imaging this across time, and there's a little arrow shown there that shows a new dendritic branch forming, contacting an existing dendritic branch around 12 minutes and making a turn, which is more obvious at the end at 28 minutes. This prevents our dendrites from overlapping. Now whenever you have mutations in furry or tricornered, that would be panels B, C, and D, you'll notice quite a bit of overlap in these dendrites compared to the wild-type neuron shown in panel A. In panel A, we see a good degree of dendritic tiling. They're not just bunching up over each other, and that's because when they encounter a like dendrite, they'll turn. So if I were to have a new branch here come off and encounter the original, it's going to turn away to make sure it doesn't just grow over the original dendrite. That's not occupying any new space, so it's not really adding anything to the ability of that neuron to connect with its partners. Now, how does a neuron know its dendrite from its neighbors? And that's of course going to depend on surface proteins. So in mice, we think it's going to be different forms of protocoherins. These are just cell adhesion molecules. We have a pretty good understanding in Drosophila. So DSCAM1, or Down syndrome cell adhesion molecule 1, is alternatively spliced at a few different locations. So they're color-coded there. There's a few exons that are going to be alternatively spliced, where they can select one of a few or up to many different versions. And through this alternative splicing, which is going to be random, 38,000 different isoforms can be made. So the odds of you expressing the same DSCAM1 as your neighbor would be 1 in 38,000. So not very likely. So if you bump into a dendrite that has the same cell adhesion molecule as you, you know it's you. So you can turn away from that. we're not going to tolerate growing dendrites from the same neuron into the same location. And if you look here, when they knock out DSCAM1 in this Drosophila dendritic arborization neuron, notice all the little red arrows. That's showing you crossing of dendrites. It can't tell its dendrites from its neighbors, so it tolerates that overgrowing. It can't tell self. Now, how complex a neuron makes its dendritic arbor really depends on what types of genes it expresses and how well it's able to grow and branch its cytoskeleton. There's a few transcription factors that are very important in Drosophila. There's likely to be far more than that in mammals. We haven't worked out the details like we have in Drosophila, so we'll look at those. So the neurons that have been studied most extensively are the dendritic arborization neurons, which are just some sensory neurons. They can have one of 4 different classes, going from simplest to most complex. So class 1, simplest; class 4, largest, most complex. And which class they fall into depends on which transcription factors they have. So we'll go through a few of those here to get some idea of how different transcription factors can create different complexity. So, the first one is cut. Cut is going to promote the complexity of dendrites. So you'll see higher levels of expression in more complex dendritic arborization neurons. So we'll see more branching, for example. So we're going to see an overall increase in the amount of dendrites. Not surprisingly, if you were to decrease cut expression, so have some sort of point mutations or remove it, you're going to see a decrease in the complexity of dendrites. So compare the top, panel A, to the bottom, panel D. Here we've got normal cut. On the bottom we have a loss of function mutation. So cut doesn't function as well, and notice the decrease in the amount of dendrite. Overexpression of cut in those less complex neurons increases their complexity. The next transcription factor is NOT. What NOT is going to do is suppress the formation of dendritic FILOPODIA. Now they're also called dendritic spikes. I'm going to avoid that term though, because dendritic spikes I would like to be reserved for the action potentials that are generated in dendrites. So these would be the dendritic FILOPODIA. So those class 4 dendritic arborization neurons don't have a lot of dendritic FILOPODIA, and that's because they have high levels of NOT expression. The class 3, on the other hand, have low NOT expression and therefore form those little dendritic FILOPODIA. So if you increase NOT expression in class 3, they look something a little more similar to class 4. You see fewer dendritic FILOPODIA, and in this case an overall increase in the amount of dendrite. Abrupt is the transcription factor that, as the name suggests, decreases the complexity. So this is the counterpart to cut. Rather than increasing, we're going to be decreasing complexity. So the class 1 dendritic arborization neurons, they're going to have higher levels of abrupt expression, and that's why they're simpler. So class 1, if you remove abrupt, they become more complex. If you express abrupt in class 4, their dendrites become simpler. So here we can see expression in class 4 neurons. Normal on top, panel A and B, just looking across time, looking over development. C and D is showing you with misexpression of abrupt in these would-be class 4 neurons, but no longer do they look like class 4 sensory neurons. and there's large areas that are no longer covered with dendrites because the genetically encoded complexity has been cut short by ABRUPT. Alternatively, if you have loss of function mutations in ABRUPT, then your class 1 dendritic arborization neurons appear more complex, so they have a greater amount of dendrites. Now, the master regulator of this expression is spineless. So we think spineless is going to act upstream of all of these to allow their expression and then create the unique dendritic phenotypes that we see. So without spineless, you don't have your unique either high or low levels of complexity, presence or absence of dendritic filopodia. All of them kind of look the same. Class 1 and 2 become a little more elaborate. Classes 3 and 4 have slightly less elaborate dendritic arbors. So this class 2, you'll see, wild type on the left there, a spineless mutation on the right. Notice the increase in the amount of dendrite. Compare this to a class 4 dendritic arborization neuron, where we see a decrease in the amount of dendrite, or a decrease in their complexity. So we genetically encode how complex the dendrites are. And this is why pyramidal neurons are going to have a certain shape to them, and stellate neurons will have another shape as well, because how elaborate their dendrites are is based on what transcription factors they have. Now the maintenance of these dendritic fields or dendritic tiles that we set up, that's going to depend on surface protein interactions. So, TRICORNERED, which is pretty important for turning, is going to be stimulated by a surface protein called HIPPO. So, how is it that a dendrite knows that it's encountered its partner? Well, it has a surface protein that binds to some other surface protein. HIPPO is going to be the ligand. TRICORNERED is going to cause turning. For the maintenance, HIPPO is going to interact with a different surface protein, in this case, warts. So HIPPO is going to bind to the receptor warts. And what warts is going to do is help preserve our dendrites. Doesn't seem to be too important for the formation. Wart is going to be the receptor that HIPPO binds to. And this is going to then preserve our dendrites. We don't think that it really plays a role in making them. TRICORNERED, we think, helps a whole lot with the actual dendritic tiling. But if you were to impair the function of warts by either knocking it out or knocking out SALVADOR, that's its adaptor protein, you lose the ability to maintain your dendrites. So wild type is shown on top. Here you can see the formation of those dendritic tiles over about 3 days. In a warts knockout, we create our dendritic tiles. Everything seems okay about 2, maybe 2.5 days later. But at about day 3, the dendrites are lost. So we need HIPPO interacting with surface proteins to help us create our dendritic field, but then also to maintain it. So what HIPPO-warts interaction is doing to maintain our dendrites is modifying gene expression. What we're going to do is act through the POLYCOME repressor complex. So the POLYCOME genes are transcription factors that are going to be expressed whenever we have activation of HIPPO and warts. So we'll preserve the dendrites through POLYCOME repressor complex. There's 1 and 2. And they do basically the same thing, a little bit different. the polycomb repressor complex 1 is going to repress expression by compacting chromatin. So the DNA and protein there are going to tighten up so it's more difficult to read. We'll also promote tightening up by methylating histones. Again, compacting chromatin. What these do is decrease gene expression for the targets. So, in this case, our polycomb repressor complexes are going to modify chromatin, cause it to compact up, so we decrease expression of the targets. And in this case, it's going to be different HOX genes, ultrabithorax in particular, for our dendritic arborization neurons. What ultrabithorax does, UBX, ultrabithorax. So great name. What this does is decrease complexity. So homeobox genes are a large group of transcription factors that affect development. They control the development of different parts of the body. So in this case, what ultrabithorax does is create kind of simpler dendritic arbors. It causes a culling of dendrites, so removal of dendrites. So in order to maintain our dendrites, what we have to do is make sure we keep ultrabithorax expression low, so that we don't remove our dendrites. You can see what happens here if we overexpress ultrabithorax in a class 4 dendritic arborization neuron. So panel G, wild type, look at panel H. We decrease the number of branches and the overall amount of dendrite. There are other HOX genes that are involved here. So collectively, those HOX genes are going to decrease dendritic complexity, and what Hippo-Warts interactions do is stimulate the polycomb repressor complex so that we don't create those HOX genes that simplify the dendrites, and we keep them stable. Now the development of dendrites is going to be guided by extracellular cues as well. So we have endogenous genetic programs that are going to create more or less elaborate dendrites. Then we'll have surface protein interactions that help maintain those elaborate dendrites. But the movement of dendrites into different positions is going to depend on extracellular cues, just like axons. So when neurons are first putting out their little neurites, they don't know if they're dendrite or axon. They have to make that decision based on where they are. So usually there's some kind of gradient to help identify what's up, what's down. Maybe I want to grow my dendrites up and my axon down if I'm in the cortex. There's no point in growing my axon up toward the surface. There's nothing to talk to there. If I'm going to project downward to the spinal cord, I need to project my axon downward. I should grow my apical dendrite up, maybe spread a few basal ones out. So we're going to be riding along similar gradients, for example the slits and SEMAPHORINS. Those will repel our axon, push it downward, but what they'll do is help promote growth of dendrites upward. So by having one or a couple of gradients here, we can both guide axons and dendrites so we don't put them in the same spot. One neuron is going to want to project its axon away and grow its dendrites locally. So here we can see a SEMA 3A gradient here in the cortex. So the top would be more the apical surface, and on the bottom ventricular. SEMAPHORINS are going to repel axons, but what they'll do for dendrites is help them grow. And what they'll do is help adopt a dendritic phenotype. So anything that does grow upward, it's going to become dendrite. They won't decide to become axon. It'll be that neurite that's exposed to the lowest levels of SEMAPHORINS. So this helps the neuron make the decision which one of these neurites should be an axon. So in the case of the cortex, that neurite that's growing away from the peel surface, where you have the highest levels of SEMAPHORINS. And what SEMAPHORINS do is recruit a soluble guanylyl cyclase into the dendrites. Guanylyl cyclase makes cyclic GMP, as the name suggests, and that's going to induce a dendritic phenotype by activating cyclic GMP targets like protein kinase G. So, neurons have no idea which way to grow their dendrites and axons. They just grow them outward, and then the environment helps them make the decision. Up here at the top, where I have more semaphorin, that'll be dendrite. That thing that I grew downward, that's going to be my axon. Here we can see the effects of semaphorins on axon growth. So what they did was take a little explant of cortical neurons and put them on top. So we're looking at a little cortical surface there. So they take a bit of cortex. That's going to create your semaphorin gradient. And remember, it's going to be high to low. And they put some neurons in different spots. So when they put a little explant right here, there's definitely a semaphorin gradient. All the neurons that are growing here, we're going to see dendrite more upward, and we should see very few growing downward. So if you look in panel panel F1, that's control. Notice almost every dendrite is growing upward. That makes sense. There's more SEMA3A up there. Whenever they apply SEMA3A in the bath, that's going to be number 2 down there below. What they did was just bathe this whole slice in semaphorin. So now there's no gradient. What we would expect is that dendrites grow in all directions, and that's exactly what they saw. The quantification is in part G on the bottom. So you'll see under control, most grow upwards. When they bathe the whole slice in semaphorin, they grow in all directions. So when there's no gradient, it doesn't know which way to put its dendrites, and it grows. Each neuron grows equally in all directions. Now on the on panel H there, what they did was take some HEC293 T cells, have them expressed semaphorin. So what they did was flip this gradient. Now what they're doing is creating their own unnatural semaphorin gradient. So when they grow neurons here, what we should expect is that their dendrites are going to tend to grow down. So in part 1, when they don't have the HEC293 T cells express anything at all, what they see there is still mostly there's some upward growth. It's not as good as what you see in part F though, because we're not really getting much of a semaphorin gradient. When they cause semaphorin 3A expression in those 293 T cells there, those HEC cells, now most of the dendrites are growing downward toward that semaphorin. So look in panel I. Control, basically no semaphorin gradient, because we're away from that peel surface. When we have expression of semaphorin with that explant near the ventricular zone, and we flip the gradient, now most dendrites are growing downward. So this is how neurons solve the problem of knowing where are they. There's again extracellular cues that create gradients, and that's what tells them. Slits are also going to be important for dendritic development. So remember that slits are going to be little soluble proteins found in a few distinct locations. And when slit binds to roboreceptors, robo1 and 2 that is, it repels the axon. It's different in dendrites. Slits are actually going to promote dendritic growth. We can see that here in these data. Control on top, bathed in with some slit on the bottom. Notice the increase in dendrites. So what this does is cause cells to grow their dendrites in one area and grow their axon away from that area. When you apply slit, regardless of whether it's a pyramidal cell or a stellate cell, you see an increase in the amount of dendrite in terms of length, branch points. So you increase complexity. You use the same gradient to grow your dendrites and push your axon away. Neurotrophins still increase growth. because they're still going to lead to protein synthesis. So they help axons grow, they also help dendrites grow. The way that they do this is by stimulating MAP kinase and PI3 kinase signaling pathways. So as long as you have the appropriate neurotrophin receptor, you will have an increase in protein production and you will grow, both dendrite and axon. What we're doing there is stimulating the release of RNA from RNA binding proteins. So we're going to release those local RNAs in the dendrite, and we're going to allow for the formation of functional ribosomes. So we're going to inhibit proteins that hold the ribosome in its kind of separate subunits. So those eukaryotic initiation factor binding proteins that keep the ribosome from assembling, we inhibit those. You can see the little cartoon here. Both PI3 kinase and MAP kinase are going to accomplish the same thing. PI3 kinase is going to act through mTOR, the mammalian target of rapamycin. But what they're both going to do is facilitate the formation of ribosomes and the release of RNA, and we need both of those. So that we let go of the local RNA, we now have a ribosome, then we create protein. And what that protein is going to do is help create the cytoskeletal components that we need in order to actually grow new dendrites or axons. We're also going to regulate the stability of those cytoskeletal elements. It doesn't do you much good to make a bunch of actin if you can't make actin filaments. So those MAP kinase and PI3 kinase dependent signaling pathways are going to stimulate Rho GTPases. And collectively what the Rho GTPases are going to do is cause polymerization of cytoskeletal filaments. There's a few different master regulators. There's Rho A, RAC, and CDC42. They're a little unique, as you can see in this cartoon. They have slightly different targets, but overall what they're going to do is promote complexity of the cytoskeleton and promote stability of the cytoskeleton. They'll facilitate growth. They'll do it a little differently in different parts of the cell and different types of cell, but that's their general function. Now, if you were to knock out RAC, for example, that's going to decrease the stability of your cytoskeletal components. It seems that RAC is playing a bigger role in the dendrites than the axon, because if you look at RAC1 knockouts, we see a loss of dendrites, not so much axons though. But those different surface proteins that were mentioned here, those are going to be receptor tyrosine kinases. Those are going to help promote dendritic complexity by stimulating MAP kinase and PI3 kinase dependent signaling so that we can create and stabilize cytoskeletal filaments. Wnt also, of course, plays a role here, just like it does in axons. So Wnt will bind to that frizzled receptor. There's a scaffolding protein, disheveled, which is then stimulated. And disheveled is going to stimulate Rho GTPases, in particular RAC. So disheveled and RAC interact directly. Whether we stimulate RAC or some other Rho GTPase, that then causes polymerization of the cytoskeleton. If we were to mutate disheveled or knock it out, we'd see shorter, less complex dendrites. So have a look at the top row. That's a couple of wild type neurons. In the bottom, when you knock out disheveled, less complex dendrites. Or if you mutate RAC, very bottom row, so that RAC N17. We're not really seeing any dendrites. We still see an axon, but the dendrites are very, very, very short. That immunoprecipitation inset is just showing you that RAC and disheveled are pulled down with one another. So when you immunoprecipitate disheveled, you can detect RAC. That suggests that they have a direct or pretty darn close to direct interaction. Calcium-dependent signaling is of course going to affect dendritic growth. Calcium is still a secondary messenger. So when neurons are active, that's going to help promote the formation of more complex dendritic arbors. And there's a couple of different calcium-dependent kinases that are going to promote this. CAM kinase 2, for example. what that's going to do is promote actin growth by inhibiting cofulin. Cofulin is an enzyme that cuts up actin. So by inhibiting an inhibitor of actin growth, gamma kinase 2 promotes actin growth. Gamma kinase 4 is going to act mostly through CREB, and it's going to increase the expression of candidate plasticity gene 15. Candidate plasticity gene 15 is a cell surface protein that appears to interact directly with elements of the postsynaptic density, such as AMPA receptors. And what it does is lead to the formation of larger and more numerous postsynaptic sites. Not surprisingly, you see an increase in the amount of dendrite. So compare the top cells. There's 3 different cells followed over 3 different days, so you get a 3 by 3 array. On top, they put on a little beta-gal, doesn't really do anything. In the bottom 3 cells, they applied the candidate plasticity gene 15. When they transfected them to express candidate plasticity gene 15, these cells then inserted into their membrane, and that allows for the formation of more complex dendrites. So if you just compare the top to the bottom, you should see the general trend there of more elaborate dendrites. Now, you will in some cases find hotspots for synapse formation, where the axons and dendrites of 2 target cells have to find one another. So in the case of the cortex, not really the case. You'll see synapses kind of along the dendrites, but in places like the retina, you definitely see this. There'll be distinct layers where we see synapses forming. Most likely what's going on here is that there's a semaphorin gradient where it's higher in the internuclear layer. That's going to allow axons and dendrites to know where they are. So the axons of the neurons in the internuclear layer, those are going to grow downward. So very similar to what we just had here. But in this case, we're going to create a semaphorin gradient. And in those cells in the internuclear layer, they're going to want to grow their axon downward away from that. And those down in the ganglion cell layer, well, of course, they're going to want to grow their dendrites upward. Now, where they meet, of course, depends on the type of cell that's there, and which partners they bind to depends on what surface proteins they have. So here it looks like homotypic interactions between axon and dendrite allow partners to find one another. So there's those SDK surface proteins, there's the DSCAMs. Homotypic interactions allow the pre- and postsynaptic neurons to stick to each other. So for example, if they both express DSCAM, they're going to stick and they're going to form synapses. Where they meet up depends on how responsive they are to that semaphorin gradient, so you'll find some synapses here, others might be a little lower in a different layer, but we do see distinct layers of synapse formation. That's likely a combination of this semaphorin gradient and then specific surface proteins that allow one neuron to stick to its partner, but not the others. Now the last thing that we need to discuss is the difference between the microtubules and axons and dendrites. After the cell decides which neurite becomes the axon, then it goes about modifying those microtubules. And what it does is specialize them for long distance transport. Now, throughout neurons, you're going to see neurofilaments, those are the intermediate filaments. You're going to find actin filaments, and you'll find microtubules. So neurofilaments are just kind of like rebar. You find a lot of these in neurons. They help give them stability. They're not really useful for transport though. Long distance transport is all about microtubules. They're a little bit bigger than the intermediate filaments. And they also have polarity. And that's what's going to allow them to actually ship stuff in a given direction. One unique thing about the microtubules of neurons is that they're not held to that microtubule organizing center like they are in most cells. In neurons, the microtubule organizing center is just a place to start growing your microtubules, and then you cut them off and move them elsewhere. So we see little discontinuous collections of microtubules. That's unique. Another unique aspect is the mixed polarity we find in dendrites. In every other cell, plus N goes toward the periphery, and this is true for axons. In dendrites, though, plus can go away and plus goes towards the center. That's the mixed polarity there. If you're wondering how do they know how to find the right direction in dendrites, I have no idea. That's still a mystery. Also distinguishing dendrite from axon, besides the mixed polarity, would be the microtubule-associated proteins. And these are exactly what they sound like. They're proteins that associate with or bind to microtubules. In the dendrites, cell body, you see MAP2, microtubule-associated protein 2. In the axon, you find tau. They bind to microtubules, they help stabilize them, and you see different proteins in different locations. MAP2 is stained blue in this case, tau is stained red. So you can see the dendrites, cell body, those are blue. You can see the axon clearly as red. The microtubules themselves are also distinct in axons versus dendrites. So in axons we're going to see removal of the C-terminal tyrosine residue, and that's going to leave a glutamate. So you can have different antibodies to recognize this detyrosynated alpha tubulin. What detyrosynation does is promote interaction with kinesin. Kinesin is a motor protein that's going to allow for plus N directed movement, thus that anterograde axonal transport. This is a good thing because we may have to transport intracellular components over a great distance, depending on the type of neuron we're talking about. So if we innervate the periphery somewhere far away, we have to transport everything in that axon down the entire length. So by allowing for more stable interactions with kinesin, that can help speed up the rate of anterograde trafficking. In axonal microtubules, you're going to see a whole lot more detyrosynated tubulin. There's also the addition of acetyl groups onto the tubulin subunits. So when tubulin subunits are acetylated, this also promotes interaction with kinesin. Not surprisingly, microtubules in the axons are going to be more highly acetylated, again allowing for more stable interactions with kinesin, because we have much further to transport our goods. Okay, let's take a look at the data shown over in this figure. What they're looking at is the amount of tyrosynated and detyrosynated tubulin. So you can look at the actual data. Those would be the green and red signals, tyrosynated versus detyrosynated tubulin. If it's yellow, that means you have exactly the same level. If it's red, that suggests that you have more detyrosynated tubulin. So look at the plot on the bottom. And what this will show you is that in the axon, compared to the cell body and dendrites, we're seeing more detyrosynated tubulin. So this will be my green in this case. So you'll notice what they're doing is just looking over a distance. So they're following, let's say, down the axon. This will be at the soma, and this is down the axon there. In the axon, once you exit the soma, you'll see that there's an increase in the amount of detyrosynated tubulin compared to your run-of-the-mill tyrosynated tubulin. In the dendrite and cell body, if you'll go to the image to the right of that, so the bottom right, you'll notice the signals look basically the same. There's not an increase in the amount of detyrosynation. Two advantages to this. It's easier to transport stuff down the axon, which is much longer, and it also prevents kinesin from sticking to tubulin until it's been modified. So it does a better job of sticking to modified tubulin. That is tubulin found in stable microtubules. looking at acetylated tubulin here. This is just at different stages, so stage 2 on the right there, that's before it has made the decision of which neurite is its axon. Stage 3, it's making the decision. It's done a pretty good job in most places. Has a good idea. It looks like there might be, there's one main axon in part B there. You can see maybe a minor neurite that hasn't quite decided yet. But comparing stage 2 to stage 3, when you look at the amount of acetylated tubulin, it's kind of uniformly distributed before the decision is made. But once we've decided which neurite is the axon, that's where we find our acetylated tubulin. Alrighty, and that does it for dendrite development. If anything is tricky or unclear, use the questions box so we can have a more productive chat in class. I'll see you later.